To all the students out there of BPC second year, here NEET World has got a great offer for you to extend the support not just for MCET and NEET but also for IPE. So, to access the complete free materials from NEET World, you have to do few things beta. First thing, you follow us both on our Telegram channel, that is NEET World 25, and NEET World 25 Instagram channel. Follow us both on the social media platforms, and then go to Play Store, install the app MedWorld, then just say hi in chat box, just say hi in the chat box, and you will get the complete soft copies of 4 books, botany, zoology, physics, chemistry, where in each book very important long answers, short answers, VSAQs are given. Complete blueprint with weightage chart is provided. Simple and easy to solve and understand type of answers are given. Specifically from botany, though, I have also given you the flow charts and mnemonics for long answer type of questions, where you can accurately present the answer in a systematic way. Okay, so as such the content is delivered beta. This is very useful to all the students of AP and Telangana board who are appearing for the IPE exams this year. Okay, so just utilize this offer. Okay, and today we are here in what discussion? We are yet to start with plant growth and development, right? Plant growth and development, we have to start short answer type of questions. So here we go. First question is on the seed dormancy, which is asked in 2 years. If you can see, March 2023 and March 2017, 2 years the same question has appeared, to write a short note on seed dormancy. For this, what you have to do beta? First and foremost thing, you have to explain what is seed dormancy at the beginning. Just give out the definition in a proper way, and then please provide the causes and types of seed dormancy. What are the causes of seed dormancy and what are the types of seed dormancy? Last is significance of seed dormancy. This is the way you have to frame out the answer. So look at it first. Write short note on seed dormancy. Seed dormancy is a condition in which a viable seed, okay, a viable seed will fail to germinate. This is due to unfavorable conditions. Okay, even under favorable conditions, the seed will be fail to germinate. Usually this happens in unfavorable conditions, but sometimes seed dormancy is seen even in the favorable conditions, such as suitable temperature, moisture and oxygen. Such condition is known as seed dormancy. It occurs due to certain internal physiological or structural barriers within the seed. Okay, either external barriers or internal barriers will prevent the seed from germination. External barriers is when the environment is unfavorable, be it in unfavorable temperature, moisture and oxygen. When external barriers are not there, even in favorable conditions, if seed is unable to germinate, that is because of internal physiological and structural barriers. Now you see what are the causes and types of seed dormancy. First thing, due to hard seed coat. If seed coat is hard, then it is difficult for epicotyle and hypocotyle to burst out opening the seed coat. Yes or no? First is due to what? Internal barrier, that is due to hard seed coat. Okay, this is due to hard seed coat, which is physical dormancy. This is addressed as what ma? Physical dormancy. Here you see, in some seeds the seed coat is hard and it is impermeable to water and gases. This is impermeable to water and gases, which prevents the germination. For example, if you see members of Fabaceae, for example bean and pea. Fine, right? Next you see dormancy can be broken by what? The process called as scarification. What is scarification? A process of mechanical or chemical weakening of seed coat. When seed coat is tough, that is an internal barrier for seed germination. Yes or no? What is scarification? We are weakening the seed coat so that seed coat can burst out open easily and seed germination will be possible. So that is scarification. Second major point is what? It can be due to chemical inhibitors, which is addressed as physiological dormancy. Okay, some seeds contain chemical compounds which inhibit the germination even when the environmental conditions are favorable. For example, if you see tomato and other Solanaceae members, they contain what? Germination inhibitors within them. Okay. Third is what? This is due to immature embryo, which is morphological dormancy. Okay. This is immature embryo, which is what na? Morphological dormancy. Okay. Morphological dormancy. Here we go with morphological dormancy. In certain seeds, the embryo is not fully developed. Okay. Embryo is not fully developed here in few seeds. At the time of seed dispersal, so the germination is delayed until the embryo matures. Until the embryo will mature, the germination is delayed. For example, if you see here, ranunculus and buttercup are the best examples. Okay, ranunculus and buttercup. Now here we see about significance of seed dormancy. What is the significance of seed dormancy? So we have looked up to the causes. One is physical barrier, second is chemical, and third is physiological. Three types you have to mention, ma'am. After this, what is the significance of seed dormancy? It helps the seed to overcome the unfavorable conditions, be it drought, frost, etc. so that seed is viable even under the unfavorable condition. That is one major benefit about seed dormancy. Second is what? This ensures uniform germination during favorable seasons. Okay, this will ensure what beta? Uniform germination during favorable seasons. And then this will prevent pre-harvest sprouting. Okay, this prevents out what? Pre-harvest sprouting of seeds in crops, and this will aid seed dispersal and survival over long periods. Fine, right? This will aid, or aid means this will support the seed dispersal and survival for longer periods. Clear till here? So, how you have to explain if at all a short answer type of question comes on seed dormancy? First is give out the proper definition of seed dormancy and then explain about types and causes of seed dormancy. It can be due to physical dormancy, physiological dormancy or morphological dormancy. Lastly, give out the significance of seed dormancy. That's it. Okay, this is how you have to present the short answer, okay, on seed dormancy, right ma? Next, these are the points to be remembered for dormancy. Next is write a short note on agricultural or horticultural applications of auxins. We have to give out the agricultural or horticultural applications of auxins. First, this is appeared in May 2024, June 2023, May 2022, March 2020, May 2018 and March 2018. Fine, right? So, here we go with first answers. How you will give out the applications, agricultural, horticultural applications of auxins. Means you have to present out both natural and synthetic auxins here. Look into the applications beta. First, auxins comes under growth promoting hormones or growth inhibitory hormones, that you have to explain in the foremost point. Auxins are plant growth hormones that play a vital role in cell elongation, apical dominance, root initiation, fruit development and abscission. Okay, this plays a vital role or a major role in cell elongation, apical dominance, root initiation, fruit development and abscission. Remember all the major points. They are extensively used in agriculture, horticulture to improve growth, yield and productivity. It improves all the things, that is growth, yield and productivity. Fine, right? So, here auxins are what? Growth promoting hormones. They will they possess all these activities in the plants, and this is how they are useful in increasing the growth, yield and productivity, okay, as they are growth promoting hormones. Okay. Now, you see what are its agriculture and horticultural applications. Applications of auxins. Every point is important, ma. Present the answer in the more accurate way which will impress the examiner. Okay. More accurate way you have to present. If you give out your answer paragraphs without any major points in that, without any gunshot points in that, nobody will look into your answer. So whatever answer you are providing, please provide it in the point wise manner and provide most accurate information. What is exactly that is asked in the question, you have to present it accordingly. Got it? So here you see first is what ma? Root initiation in stem cuttings. Auxins promote the formation of adventitious roots. They will promote the formation of adventitious roots in stem cuttings. Next, this property is widely used in vegetative propagation of plants. Okay, this is widely used in vegetative propagation of plants. Best example for root initiation in stem cuttings if you take, that is application of indole acetic acid, indole butyric acid and naphthalene acetic acid in stem cutting regions of sugarcane, coleus. This will induce what? Root formation in stem cutting regions. Fine, right? What you have to remember here, what type of auxins that we are using to initiate rooting in the stem cutting regions and what are the examples of this feature. Fine, right? You have to mention what type of auxins we are using and what are the exact examples of root initiation in stem cutting regions. This is what is important. Okay, this and all you can present in your own terminology as well, but then mentioning these two are important. Next, promotion of flowering. Promotion of flowering. Certain auxins will promote flower initiation. Okay, they will promote flower initiation and uniform flowering in the plants. For example, if you see spraying of auxins, this will induce what ma? Spraying of auxins will induce flowering in pineapple plants for synchronizing fruits, for synchronized fruiting. Fine, right? So here auxins are promoting flowers, examples is what? Pineapple. Fine, right? Next, prevention of premature leaf and fruit drops, that is it will prevent the abscission of young fruits, young flowers and young leaves. That also you can mention, it is your wish. You can write it out in this way or you can also mention like it will prevents abscission of young fruits, flowers and leaves. Fine, right? And then here you see auxins will prevent early abscission of leaves, fruits, thereby it will improve the yield. Next, however, they help in natural abscission. Natural abscission is observed in older and mature leaves and fruits. In older and mature leaves and fruits, you can see natural abscission. Okay, that is also done by auxins. Auxins will prevent premature abscission and auxins will promote mature abscission. Okay, those fruits which are ripened, those leaves which are matured, those will fall off from the plant. And then this the control of apical dominance. How can we control apical dominance? Apical dominance is a feature where auxins will promote the growth of apical buds and inhibit the growth of lateral buds. So apical dominance is controlled by decreasing the auxin levels and increasing cytokinin levels. Fine, right now? So here you see decreasing auxin and increasing cytokinin level. Auxins produced at the shoot tip will inhibit, this will inhibit lateral buds. This will inhibit lateral buds, maintaining what? Apical dominance. This will maintain apical dominance. When the shoot tip is removed, or decapitated, lateral buds will grow actively. Okay, lateral buds will grow actively here. The principle is used in what? Tea plantations and hedge making to promote bushy growth. This will promote what? Bushy growth. This is what you will see it in the control of apical dominance. Next, induction of parthenocarpy. How can you induce parthenocarpy, which is seedless fruit formation? Okay, right, ma? This is what? Seedless fruit formation. Auxins will stimulate, this will stimulate the development of seedless fruits. This will stimulate the development of seedless fruits without fertilization. For example, here you can see tomatoes and bananas can produce parthenocarpically using what? Auxin sprays. Clear till here? This is what? Induction of parthenocarpy. Next, use as selective herbicides. Okay, use as what? Selective herbicides. Synthetic auxins such as 2,4-D, that is 2,4-dichlorophenoxyacetic acid, is used as weed killer, okay, and they will kill dicot weeds, but it will not affect monocot crops like wheat and rice. Okay, this will not affect monocot crops like wheat and rice. If you see the examples over here, 2,4-dichlorophenoxyacetic acid is used to control weeds in wheat and maize fields. Clear, right? Clear till here how it is used as selective herbicides, how auxins are used as selective herbicides. Is this clear till here, ma? Okay, next you see weed free lawns, which is very important. Auxins will promote weed free lawns. How? Auxin herbicides like what? 2,4-dichlorophenoxyacetic acid, when it is sprayed on lawns to destroy broad leaved weeds, especially dicot weeds, without harming grass. For example, if you see here, gardeners will use 2,4-D to maintain clean and weed free lawns. First thing what I tell you is, you have to remember how that you have to mention out that auxins are growth promoting hormones, and then you have to start on giving out the applications. What are the major points you have to remember? First thing is root initiation in the stem cutting regions. What auxins we are using? IAA, IBA and NAA. On what plants like sugarcane and coleus. Second is promotion of flowering. Example you have to remember is pineapple. Third, prevention of premature leaf and fruit drop, that is by what, ma'am? Which is preventing abscission, early abscission. Next, control of apical dominance, that is by removing the apical regions, which is known as decapitation, which is observed in tea plantations and hedge making. Fifth is what? Induction of parthenocarpy, that is promoting seedless fruits. Then you see use of selective herbicides like 2,4-D, and then weed free lawn preparation, that is also by 2,4-D. These are 7 applications that you have to fix it in your mind. Okay, this will award you complete marks, ma'am. Okay, complete content is provided, but present it in a systematic way. For every question, for every concept, you have to remember the content in a structured framework. Okay, then only you can present it well. Fine, right? And don't just mug up the questions. Never mug up the questions. Learn the answer in a structured format, so that content is useful for you people to present your answer both in the subjective form or objective form. Learn the content properly. Fine, right? So next question you see, write physiological reasons for gibberellins in the plants. Write physiological responses of gibberellins in the plants. That will be a third. And then physiological effects of cytokinin, and then physiological effects of ethylene. Yeah, 3 physiological effects you have to note. So when plant growth and development comes into the picture, you have to highlight on what? Plant hormones or plant growth regulators, both plant growth promoters and plant growth inhibitors. All are important. Plant hormones are important not just for your IPE, it is important for your MSET and for your IPE as well. So look into that more carefully. Okay. So here we go with this. Write the physiological responses of gibberellins in plants. What are the physiological responses of gibberellins in plants, which is appeared in Feb or March 2024 and May 15 and March 2015. So 3 times appeared in your previous year paper. So any of the papers you see, there will be one question on phytohormones. So it is important, just go through it carefully. Here we go with gibberellins are plant growth hormones that regulate wide range of developmental and physiological activities in plants. Okay, they will regulate what? Wide range of developmental and physiological activities in plants. They mainly promote what? They mainly promote stem elongation, fruit growth, seed germination and flowering. Okay, they will mainly promote what? Stem elongation, fruit growth, seed germination and flowering. This is being given like physiological responses of gibberellins. First you have to mention out clearly in the beginning only, like what are the exact functions of that particular phytohormone. Then go on elaborating for each type of function. What are the physiological responses? You have to elaborate it in this way. First you see stem and internode elongation. That is the major role of gibberellins. Cell elongation or internodal elongation is the major function of gibberellins. Here you see gibberellins will stimulate cell division and cell elongation in stems and internodes, leading to an increase in plant height. This will increase what? Plant height. For example, here you see they are used to increase length of grape stalks, they will increase length of grape stalks and they will improve cluster appearance. Fine, right? So increase in the length of grape stalks, this is the mandatory example that you have to give for stem and internode elongation. Second is fruit growth and shape improvement. How can you improve the shape and fruit growth? That is also by applying the gibberellins. How? The stalk size, main stalk size of the pedicel, it will be increased. The entire flower turns to what? Fruit. Ovary size will increase. The stalk which is bearing the fruit size will be increased. As such, the fruit will get a definite shape by the application of gibberellins. So here we go with application of gibberellins to young fruits will cause what? Elongation of the fruits, and this gives what? Better shape formation. Fine, right? Here you see example. In apples, gibberellins will enhance. Fruit size and this will improve the quality. Third major point is delay of senescence, that is aging. Gibberellins will delay aging process, that is senescence process will be delayed of both leaves and fruits. This will allow fruits to remain longer on the plant. And this is extended in the market so that the shelf life of the fruits will be increased. Okay, shelf life of the fruits will be increased because of application of gibberellins. Next is what? Increase in sugarcane yield. How sugarcane yield will be increased? By the application of gibberellins. Why? Because gibberellin application will increase the internodal elongation of the stem, where stem is the edible part in the sugarcane. As such, by spraying gibberellins on the sugarcane crops, that will increase the stem elongation and internodal length, thereby increasing the yield of the sugarcane. Clear, right? Just by application of the gibberellins, that will increase the yield up to 20 tons per acre. Okay, it will increase the yield up to how much? 20 tons per acre. Next is early seed formation in conifers. When applied to juvenile conifers, when gibberellins is applied to juvenile conifers, gibberellins will accelerate reproductive maturity. Okay, this will increase the reproductive maturity, leading to what? Early cone and seed production. This leads to early cone and seed production. Next is what? Induction of bolting. Gibberellins will promote bolting. Bolting means what? Rapid elongation of floral stalk. But this is sprayed. Gibberellins will be sprayed prior to flowering, so that when the stalk size, pedicel and peduncle size is increased, that will give rise to broader flowers. In plants with rosette plants like beet, cabbage, lettuce. Before flowering itself, gibberellins will be sprayed. This is especially useful in the crops which grow in cold climates. Clear, right? So, gibberellins will promote bolting. What is bolting? Which will increase the size of stalk, floral stalk. When floral stalk is increased, stalk size or length is increased, that will give rise to broader vegetables and broader fruits. Fine, right? This is a major application that we will use for what plants? Rosette plants like beetroot, cabbage, lettuce. Clear till here? Any doubt anywhere? Next you see, breaking dormancy. Breaking dormancy and promoting germination. How the seed dormancy is breaked and how the germination is promoted. Here we go. Gibberellins will help breaking what? Seed dormancy, and this will stimulate germination by what? Inducing the synthesis of hydrolytic enzymes. This will induce what, ma? Synthesis of hydrolytic enzymes like amylase, which will mobilize food reserves in endosperm. This will mobilize what, ma? Food reserves in endosperm. How? Gibberellins will promote the seed germination. In during the time of seed germination, in starchy endosperm, what is present? Starch is present. Starch should be simplified to simple sugars. Okay, in the starch should be simplified to what, beta? Starch should be simplified to simple sugars by the enzyme amylase. The amylase synthesis is triggered by gibberellic acid. As such, gibberellic acid will help in the promoting seed germination. Clear? So, this is how it is helpful in seed germination. So, these are the major points that you have to remember. What are those, ma? First thing, physiological responses of gibberellins. What does it include? Major thing, immediately whenever you are remembering the hormone, phytohormone gibberellins, you have to remember internodal elongation and bolting process. Third is seed germination. These are the 3 points you have to quickly remember when you are getting the term of gibberellins. So, here if you see, first is what? Stem and internodal elongation. Second will be fruit growth and shape improvement. Third is delaying senescence. Fourth will be increase in sugarcane yield. Fifth is seed formation in conifers, and then bolting. Lastly, what? Breaking the dormancy and promoting seed germination. These are the physiological effects of gibberellins. What are gibberellins? Plant growth promoters. Next, about cytokinins. Cytokinins are also the plant growth promoters. So far we have dealt about auxins, gibberellins, now cytokinins. Just go through it, ma. I will be back in a minute. Here we go. Write any 4 physiological effects of cytokinins in plants. So, the question has appeared previously in May 2022 and 2019 and 2016. Here we go with the basic explanation. Of course, for any phytohormone, in the beginning only you have to explain whether that phytohormone is a plant growth promoter or a plant growth inhibitor. And roughly you give out the physiological processes or responses that it is giving. If you see cytokinin, that is of course a plant growth promoter. What are its physiological processes? It can be growth, differentiation, delay of aging. Fine, right? Growth, differentiation and delay of aging in the plants. Here we go with the physiological effects. First is what, ma? Promotion of cell division and leaf formation. First is promotion of cell division and leaf formation. Fine, right? Here you see cytokinins will promote cell division and they will help in formation of new leaves and chloroplast. Okay, they will help in the formation of new leaves and chloroplast. This will play a key role in tissue culture. This will play a key role in tissue culture by stimulating shoot formation from the callus tissue. From callus tissue, this will stimulate what? Shoot formation. Fine, right? Callus is undifferentiated mass of the cell that you will see in the tissue culture, right? So from callus, if more auxins are applied, more auxin concentration is applied to callus, this will promote what? Rooting. More cytokinins is more cytokinin concentration is given, this will promote what? Shooting. Fine, right? Rooting and shooting. First promotes cell division and leaf formation. Next, induction of lateral and adventitious shoot growth. So cytokinins will stimulate the growth of lateral buds and this will promote the formation of adventitious shoot leading to branching in plants. Next you see ma, overcoming apical dominance, that by increasing the cytokinin concentration, apical dominance is prevented and this will promote the growth of what? Lateral buds. So here you see apical dominance which is caused by auxins is counteracted, it is counteracted by cytokinins, and then by promoting lateral bud growth, cytokinins will help plants to overcome suppression by what? Apical bud. Next, last point is delay of leaf senescence, that is anti-aging effect. This will give out what? Anti-aging effect. Cytokinins will delay aging, that is senescence of leaves by maintaining what? By maintaining protein and chlorophyll content. Okay, cytokinins will prevent the aging by maintaining protein and chlorophyll content. Most important thing is cytokinins will promote nutrient mobilization. Cytokinins will promote nutrient mobilization. As nutrients are being given or moved with the help of cytokinins, it will prevent the aging of the leaf. They will mobilize nutrients towards leaves, keeping them green and active for a longer time. So hence they are called as what? Anti-senescent hormones as well. Okay, cytokinins are also addressed as what ma? Anti-senescent hormones. So here you see what are the physiological processes or physiological effects of cytokinin. First is promotion of cell division and leaf formation. Second is induction of lateral and adventitious shoot growth. Third is overcoming apical dominance. Fourth is leaf senescence will be delayed by what? Nutrient mobilization. These points please explain it out in 8 accurate points, that will award you full marks, okay, for physiological effects, and these major points are important for your MSET and NEET as well. Okay, phytohormones are hotspots, you can say, of this chapter, very important. Here we go with what are the physiological processes regulated by ethylene in plants. Okay, physiological processes regulated by ethylene in plants. This question is appeared in May 2017. So ethylene here is a simple gaseous plant hormone. Okay, this is a simple gaseous plant hormone that will help or that plays a major role in what? Regulating growth, and this will promote ripening. This will help in regulating the growth and this will help in ripening. This will help in senescence, and this will also help the plant to survive in stress responses or in stressful conditions. It influences several physiological processes in plants. What are the physiological effects of ethylene? Important ma, just remember. Ethylene acts both as plant growth promoter and plant growth inhibitor, remember. Okay, ethylene will act both as plant growth promoter and plant growth inhibitor. Look at here, triple response in seedlings. How is it producing the triple response in seedlings? Ethylene will cause horizontal growth of seedlings, horizontal growth, swelling of hypocotyle, or you can say swelling of stem axis, and formation of apical hook in the dicot seeds. Triple response is the most unique feature that is given for ethylene. Okay, ethylene will give out the triple response, that is a major point to be discussed. What are the three responses that is shown by ethylene? You can take the example as rice seedlings. Here you can give out horizontal growth of seedlings, second is swelling of the stem axis, third is formation of apical hook. Okay ma, formation of apical hook. Then these changes will protect the shoot tip, it will protect shoot tip while emerging through the soil. Ethylene will promote aging, that is senescence, it will promote aging or senescence, it will promote abscission, that is shedding of leaves, flowers and fruits. Ethylene will promote aging or senescence, abscission, that is shedding of leaves, flowers and fruits. Next is ethylene is the most effective hormone for what ma? Fruit ripening. For fruit ripening, ethylene is the most effective hormone. It will convert starches and acids into what? Sugars, and it will soften the fruit by what? Dissolving pectin. This will dissolve what? Pectin. And this is common in climatic fruits like what? Banana, mango and tomato. Fine, right? Next you see, ethylene will enhance rate of respiration. Ethylene will enhance rate of respiration during what? Fruit ripening and process called as what? Respiratory climacteric. Okay, this will enhance the rate of respiration, where you will call it as what? Respiratory climacteric. Next, ethylene will break dormancy in seeds and buds. This will break dormancy in seeds and buds. This will promote germination and sprouting. Okay, this will promote germination and sprouting. Examples, if you see, germination of peanut seeds. Okay, germination of peanut seeds, sprouting of potato tubers. Germination of peanut seeds and sprouting of potato tubers. In deep water rice plants, ethylene induces what? Ethylene induces rapid elongation of internodes and petioles, which will enable leaves to grow above the water surface. Lastly, ethylene will promote formation of root hairs and lateral roots. This will promote formation of root hairs and lateral roots, and this will increase the absorption surface area of the roots for mineral uptake. And lastly, if you see ma, ethylene is used for what? Inducing flowering, and this will synchronize fruit setting in crops. This will synchronize fruit setting in crops such as pineapple. It also promotes flowering in mango, and it will regulate the fruit development. Despite of all these points, what are the major things that you have to explain is, first is ethylene is both plant growth promoter and plant growth inhibitor. Then about triple response, of course majorly ethylene will promote what? Senescence, and most effective hormone for fruit ripening. And the major point you have to explain is, it will increase the rate of respiration, it will act as respiratory climacteric, and it will also help in germination of peanuts, sprouting of seeds. For seed germination after gibberellins, it is ethylene which is helpful, and it will also promote what? Growth of root hairs and lateral buds. Most importantly, it will increase the absorptive surface area to increase the mineral uptake. In rice plants, if you see, it will increase the internodal elongation for what? As such, the petioles length also will be increased, which will enable the leaves to rise above the water surface. Lastly, it will promote flowering. You have to give out the examples. It will induce flowering in pineapple. It will also promote flowering and regulate the fruit development. For flowering, induces flowering in pineapple. This is the common synergistic effect which is given by ethylene and auxin. Okay. So, these are the physiological effects of ethylene. mention in 8 accurate points ma, you will be awarded with the fullest marks. You have to give out the detailed content in more accurate way, and the concept of phytohormones which you are learning here for IPE is major concept both for EAP set and NEET. It is one of the most important concept. Even if you want to analyze the previous year questions, one of the major concept is phytohormones. So, this is important for your IPE, MSET and NEET. Go through it accordingly. Okay. So, with this we have completed short answer type of questions from plant growth and development, and next we will move on to bacteria and viruses, that is microbiology, which is again the major gunshot short answer type of questions will come from both chapters. In our next class we will discuss ma. That's all for today's session. Thank you.